Now that we've looked at the big picture about kind of the arrangement of chromosomes as far as the loci on chromosomes, the concept of having heterochromatin and euchromatin and the change between those. Now remember that, as we said, heterochromatin and euchromatin are sort of a continuum. They're the ends of a continuum, and we see things going in and out. And there's some things that stay as euchromatin, some things that stay as heterochromatin, but then they may fluctuate. So now we want to focus down on actually what a gene might be. And so we're going to look at these things called open reading frames, which is another way of saying areas that are transcribed. So in this first part, we're going to concentrate pretty much on what these might be. And then in the second part of this, we'll talk a little bit about regulatory sequences and what the other parts of a gene actually are. And then we will be looking at these more in detail as we go through other lectures in the future. Okay. So what have we called a gene? Well, we called it a locus, we called it alleles, we talked about genotypes, we talked about all sorts of things about what genes are. Okay, and we've used multiple names because we want to talk about them at different times. When you say allele, you mean that you've got at least a diploid and that you've got potential variants. The piece of genetic information on this side at a locus is different from over here, but those are still genes. As I've said before, gene is a broad term. So before we actually look at the structure of a gene, it's probably important to go back and just think a little bit about this euchromatin idea, right? This idea that these light areas that take up less stain when we stay in these metaphase chromosomes are areas that stay active and represent our genes. And what we didn't mention were, yes, these are the unique DNA is the genes for the most part in some sense, but the majority of those are what are known as solitary genes. These are where pretty much you just have one copy. Okay, now you get technically two copies because you're diploid, but I mean there's only one locus. So this gene for a particular enzyme activity or whatever protein is not found in 5 places across the genome. It's only found pretty much in one place, a single copy. The vast majority of euchromatin, of this functional genes that we think of, these 20,000 genes, are considered solitary. Now they may be duplicated. There's pretty good evidence that we have 1 or 2 copies of some genes, and then they've been slightly modified. And then there's even repeated genes where they can often be tandemly repeated. And the ones you see like that are tRNA genes and ribosomal RNA genes. We'll talk more about those when we talk about transcription, and we talk about then ultimate translation where tRNA and rRNA are used. But those are other areas. Okay, but I wanted to talk about a couple of others kind of specifically. One, I want to talk about what are known as multi-gene families. Multi-gene families are where you take these original solitary genes and you copy them, and then they get dispersed out, usually across multiple chromosome locations, and they specialize into various forms. Okay, the alternative of this, which is kind of linked to it, are known as pseudogenes. Pseudogenes are these non-functional genes that probably came from these same types of duplication events, but they ended up now being deleted or dysfunctional as far as their activities. So you had these multiplications, and then some of them worked out and became specialized or different, and then some of the genes just got mutated, either the control regions or the gene themselves, so much that they're not functional anymore. So we're just going to take a quick look at both of these. We're going to start out looking at these hemoglobin genes for just a moment, and particularly because there are classically in mammals, there are embryonic, fetal, and adult, because we go through different stages and different times that we would need those particular genes, right? Well, the genes that do this are really ones that we definitely know came from duplication events. Somewhere around 450 million years ago, there was a duplication in the beta gene, and the beta hemoglobin, we have to mention, is made up of, is what we'll learn to be calling a tetrameric protein. It's a protein made up of multiple pieces of these polypeptides. In this case, it's 4 of them, and 2 of the polypeptides that make it up are the beta globin, and 2 of them are the alpha globin. And so we're concentrating right now on the beta globin part of that. About 450 million years ago, there's a duplication into the beta gene into an A and a B form. The A form continues down the line into birds. It continued on and probably into monotremes, which are egg-laying mammals. And we think that because it's definitely there, in a version of it, a similar version to what's seen in birds, is there in the marsupials, those mammals that where the young are born and come out and attach the outside of the in the marsupium. And that's not carried through, to our knowledge, in the eutherians, which are the ones with the placental mammals, right? The ones that as we are, in great apes and other things. At least it's not known to be carried through there. What is carried through for there is this beta, this other mutated form, this duplication form. That form shows up in the monotremes, okay, in the egg layers. It also shows up in the marsupials, but it shows up there after a duplication event. And then after the marsupials came off, so we're talking about millions of years ago, the split between placental mammals and marsupial mammals, we had several other duplication events. Each one of these little dots represents a duplication, so that in the eutherians and the placental mammals, we have this whole array of these potential types of this gene, these different forms. So we've just taken the same gene, we've duplicated it, and then we've duplicated it several other times. And in that duplication, we've actually specialized some of the forms. So there's an embryonic form. Keep clicking backwards. There's an embryonic form, there's a fetal form, and there's some adult forms that contribute to this structure, as well as some other secondary forms. Now, it turns out that once that gene is duplicated, it actually has ended up on a couple of different chromosomes. Now this goes back, this is not the beta, this is the original globin gene itself. It separated itself into an alpha and a beta, and then the betas themselves duplicated again to give all these other forms. But the alpha and beta forms actually are on separate chromosomes, and they're in different parts of those, okay? And there's even pseudogenes here. There are ones where we clearly see that some of these genes have been shut down, at least in human systems, okay? And of course, we have these embryonic forms, fetal form, and adult forms for all these different ones. So these all represent multi-gene families. Their existence, their forms are there, okay? Now, what we see is that the alpha part, this gene is pretty much turned on very shortly as we go through the start of fetal development, okay? And it's turned on particularly in the liver, it's in the spleen, and then as we become adults and air-breathing individuals after parturition, after birth, all of this happens in bone marrow. That's where we're going to produce the hemoglobin molecules. And this is active, but now early on, you have what's known as an embryonic form of it, because think about it, an embryo is basically this little unit that's just dividing cells. It's not physically attached, or may attach to the wall, but it's just kind of there at the moment. It's bringing in all of its nutrients from the outside, from the sac, or from some form of transmission in and out of the embryonic materials. So it's just sort of osmosis type things. It doesn't have a heart. It's not beating. Once you get a heart, and once you get these other structures and the placenta forms, then you're going along and you're actually pumping, and you're having to transfer oxygen and other things across the placental membrane, right? And then move it around. So you need to change the form, and that's exactly what happens. As far as the beta chain starts to really modify, and we see this increase, and once we start developing those organs throughout the fetal development stage. Okay? As we get closer and closer to birth, the adult form, this B form as we'll call it here, starts to take over, and you see more and more of it produced. Once we're born, we primarily have the embryonic form, okay, being produced out of the spleen, but once the bone marrow takes over, and within about 6 months, we switch over to this full adult form, which is primarily the original alpha form up here, and in the adult beta form, right? And then that's what we carry on through adult life, with some of these others being present, but in very small amounts. Interestingly, some of these embryonics, which are falling down here and decreasing over the 6 months or so, first year of life, but usually within 6 months or so, in individuals who have things like sickle cell anemia. These actually provide protection because they're not attacked in the same way that they can carry oxygen in different ways. The cells are attacked, not the protein, but they actually provide some resistance, and individuals that have a higher level of this can actually have some resistance to natural resistance to malaria that others don't. Okay? The problem though is that if you get this continue to produce, so you don't properly turn on the proper adult form of this beta protein, and you end up keeping the embryonic form going along like this. So this line would just not drop; it would carry straight across. Um, that's called fetal hemoglobin disease, and it's a fairly rare disease, but it's a problem that you don't have the efficiency of a carrying adult. Because remember, we're air breathing, right? We go from a placental exchange to when we're born, we start to have to breathe air, and we have to move it from our lungs and be more efficient carriers and transfer, in that case, of oxygen. And this embryonic form is not as efficient. So this is the disease these individuals have struggle with having oxygen levels that are high enough. Okay? Just a couple though. We want to talk about other families. One of them, a great family, is the olfactory gene. This is what's known not just as a multi-gene family, but as a super gene family, because in mammals there are greater than 1,000 genes, and they all branch back to some basic initial genes that were there. So we've had duplications all over the place to produce these. Okay, and in mice, about 20% of those genes, so mouse is a mammal, of those thousands, so a couple hundred of them are pseudogenes. They're turned off. They're there, they're hanging around the genome somewhere, but they're not functional. What's interesting about it, okay, is that if you go to humans, you end up with somewhere around 60% of it. Okay, so just think what's going on there. And there's been some studies looking at humans and their close relatives, the great apes and things. And what we see is that as you move up the evolutionary line, these red numbers represent the pseudogenes that were turned off. There were about 50 that were studied out of the 1,000 or so, and these represent how many were turned off at various times. So 5 were turned off here, 1 was turned off there, 3, etc. By the time you get to humans, there's at least 15 of these 50. So you can imagine that's representing of a large number. That's where the 60% comes from. There's this large shutdown of our olfactory genes. That's why we don't smell as well, right? So you take your dog's constantly sniffing, you don't smell anything, dog or hot, we think about their world is really this collage of smells, right, of olfactory senses for them. For us, we have smells, but we use our sense of sight far more than, and touch and things far more than we use our sense of smell. And one of the reasons we don't have it is that we have all these genes, but they've actually been turned off and modified. So they're now called pseudogenes. So this is kind of the combination of both. It's a large multi-gene family called supergene family because it's so many of them. And then for us, many of them have been turned off. So we have lots of them ending up still around in our genome, still being part of that junk, if you will. They're still there, they're still replicated and all that. They just can't function. They don't work as functional genes anymore. Okay. All right. The classical gene that we talk about, and we're going to talk about here, is a sequence of nucleotides, genotype, and it carries information that can be expressed into the phenotype. That's what a classical gene was, okay? Which meant you had to be able to see it in the phenotype, if it was in order for it to be a gene. Now, at molecular level, we know that genes are like these tRNAs and other things. Yes, it can be a gene that's going to be read and will be made into a protein in some form and make phenotype. But it can also be a gene like these tRNA genes or RNR genes or these others where it's simply transcribed. So we're going to talk about this gene in multiple ways here, and we're going to talk about them as transcribed units. But for now, we're going to concentrate on what we consider a traditional or classical gene, something that's going to make phenotype for us. Okay. If we do that, we see that human genome has somewhere between 19,000, 20,000 genes. Average size is a little over 5,000 base pairs, but they can range from about 1,200 on up to 1,500 or so, kind of on average, but then they can jump to very large, again, 100,000 or more base pairs, but somewhere around 5,000 to 15,000 is our general size. And just to remind you, we're talking about protein coding genes, right, or RNA coding genes in these range. Right now, we're going to concentrate on this protein coded genes, things that are going to make a phenotype directly, not things that contribute to the production of phenotypes as RNA coding genes tend to do, okay, or at least in the form that we're talking about, right? Okay, so here's a big chunk of DNA, right? Where's the gene? Where do we start? Where do we look for genes in this? That's not a trivial thing. Is it Does it start here and run down this direction? Where is it? Okay. Well, in order to kind of understand that and look for genes, we kind of need to understand what a gene might look like. So this we're going to call a consensus sequence of a eukaryotic gene. Okay, so we're talking about protein coding genes here, things that these open reading frames that we might work with. Right? Consensus. And that's kind of like syndrome. It means that most genes kind of have most of this, but it's a consensus. It's things will be there and some genes won't be there in others. But some things are pretty obviously there, okay? First off, to define a gene, we define a gene as a transcriptional unit, something that can be transcribed. It's a basic name for a gene, okay? So we're going to kind of make our little transcript look something like this, okay? Now, in eukaryotic genes, in protein coding genes, and as well as in tRNA and other genes, but in protein coding genes in particular, there are two basic regions within a transcriptional unit. These two regions are called exons, and the other side is called introns. Okay, I'm sure you've heard of all these, and we're going to talk about splicing and how you get rid of them and out of them. You can think about them as scenes in a movie. Okay, you go take a picture, you go to the beach or something, you take your camera out and you take pictures, but then you realize, oh, I took a picture of the wall or my foot in the car. There's no reason to keep that part. Well, that's the way these are. You've got this long film. Some of it you want to use to make protein, some of it you don't. Okay, and that's the difference between introns and exons. So let's take a quick look at those. Exons are the areas that are eventually going to be coding. Okay, so they're the area that we're going to end up using ultimately to make a mature message and therefore to be translated into a protein. Okay. The areas that we're going to leave behind, the seams of your foot or the wall or whatever you don't want to show, are going to be called intervening sequences. Okay, and these are called the introns, right? They generally don't code for phenotype, although introns can become exons. That's one of the things we're going to talk about. Remember, one of the things we have is that we have 100,000 or so functional proteins in a mammal. We have what looks like 20,000 genes. There's 5 times as many proteins as there are genes. How does that work? One of the ways it tends to work is that you can do what's called alternate splicing. We can take a message and we can use this exon, this one, and then skip over and use this one. Another much more rare, but does function this way, is that you can have introns here turn into exons. Okay, so this one right here might in fact normally be an intron and gets spliced out, but it gets kept, or part of it gets kept and becomes an exon. So that makes it that you're produce a different product as you go on. But in general, when we think of them, and for our purposes right now, we'll say that exons are the units that end up in the mature message and therefore are used in protein. Introns are things that stay internal. They don't leave the nucleus and don't become part of the actual final message. So how do we know this exists? I mean, how do we even know? Well, we know it because remember when we went back a couple lectures and we talked about these experiments where you could take DNA and you could heat them up, right? And you waited for the DNA to come back. Well, you can do the same thing, except if you throw in RNA and you heat the DNA up, RNA is single-stranded. If you heat the DNA up to make it single-stranded, these little RNA will jump in there sometimes first, right? And so you can actually line up message with the original DNA. That's one of the ways we find out where the DNA was, is by heating it up, throwing in the message, and then looking to see where it combined. And it's called annealing. And these produce these sort of odd little strands that you see. And the book has a picture like this. This is a book from various places. The web has a number of copies of it. But what you have is any place that you have this double-stranded sort of hybrid form here. So you have this area that right here, okay? What that area represents is that on one side you've got DNA, okay? Let's make a D because I can't write all that well. On the other side, you've got RNA, okay? And so they have complex, they become a duplex of RNA and DNA. And this would be an area right here, between here and right here, where there's a good match between the message, the mature RNA, and the actual DNA that it came from. And then you get these weird loops out here, and these little loops turn out to be single-stranded DNA, okay? And what they represent are places where they're not found in the RNA. So it comes along, the RNA comes along, It's doing fine, it's making up, and then all of a sudden there's a bunch of DNA down here that doesn't match. Well, it has to do something. Well, classically, it bows up out of the way. So what we have here is that any place you have a solid line, like here and right there, these are exons. And anywhere you have these bowing out regions, like right here and right here and right here, are introns. So that's how we know we actually had introns and exons, is by doing these little experiments where you just take message, purify the message up that you wanted interested in, purify the DNA, throw it together, and look for a pattern. Again, it looks a little bit messier than this. This is what it really looks like down here. But you can see where it's bowed out. There's a place right there where it's bowed out. This is certainly a large uh, area right here of introns that are bowed out as well. Okay, so you see, and they vary in size. We'll see that, and that's going to be important later on. We're going to see that we have some large chunks of intron here, and then we have some that are much smaller that are found up in here. Okay, all right, so we have these introns and these exons. How in the world does the system know what's an intron and what's an exon? Well, it turns out that the beginning and ending sequences of introns are very highly conserved. Remember, we used that term back when we talked about some of the histones. H4 histone was highly conserved, meaning that they're very contained over, in evolutionary time, and that they're very special. You know exactly when you're at the end of an intron, whichever end that you're on, by certain sequences that are found there. Okay? And you'll play with this, but just to give you an example, the part that connects between the exon and the intron up here is known as a donor site. And then the part that connects between the intron and the next exon is known as an acceptor site. Okay? And they tend to have fairly unique and fairly consistent message. In fact, you can throw a thing we do in lab is you can throw a large section of DNA in where you're trying to figure out where the introns and exons are into some programs, and those programs, computer programs, will actually search. And what they're searching for are these little motifs, something like this, CAG, GU, where the slice is between the two Gs. Or they may be looking for another splice down here. And then you'll find that there are things like pyrimidine-rich regions. There's a branch site somewhere that's important. But the point for us is that both ends of an intron are highly conserved so that the system knows that it's there. Okay? And you can actually have the splicing. So that's the way that the system is going to work. Okay? All right. So what we've done now is we've talked about the transcriptional unit. We've talked about the fact that we've got these introns and exons, and they make up what will eventually make protein, right? But what about all the other stuff that makes up a gene? There's stuff on each end, flanking regions, that's just as important as the actual messages in here, actual code, and it becomes important to look at that. So that's what we're going to do next. We're going to build the next part of this, what a gene is, in the next lecture set.